learning is normally thought of something that the brain does, but our work has shown the spinal cord has a capacity to learn, and that was something that no one thought was true. My research is aimed at trying to save the injured spinal cord and really focusing on what happens in the first 48 hours. What can we do to the patient, for the patient, as soon as the ambulance, as soon as the medevac crew arrives, to help reduce the amount of tissue loss from that primary injury. Recently, one of my students noticed that his samples appeared to be color-coded. And I was like, how could that be? Samples from animals who had received pain input after a contusion injury were tinted red, suggesting that there had been an infiltration of blood into the damaged region. Now that's important because blood is neurotoxic and will kill the surrounding tissue, expanding the region of secondary injury and impairing long-term recovery. We have provided the data to demonstrate individuals should not get an opiate like morphine soon after injury, figuring out opiates that don't hurt the spinal cord, showing, for example, epidural administration of lidocaine soon after injury can turn off the pain signals without having a destructive effect. All of that can be translated in the next 2 to 5 years. In fact, that is why Department of Defense is interested in our work, because they see it as something that can be taken to the battlefield and change the way people are treated on the way to the hospital to help spare the tissue. Texas A&M is one of the strongest centers in the area of spinal cord injury, and that would bring new research that will promote recovery after spinal cord injury.